Welcome back to MicroVerse. In the previous lecture, we discussed the bacterial cell wall and learned that peptidoglycan provides shape, strength, and protection against osmotic lysis. But bacterial cell wall are not all built in the same way. Some bacteria have thick peptidoglycan layer, while others have thin peptidoglycan layer with an additional outer membrane. This difference is also responsible for one of the most important reactions in bacteriology, the Gram reaction. So today's question is, why do gram positive and gram negative bacteria stain differently? So let's first compare their cell wall. So gram positive bacteria have a thick peptidoglycan layer, teichoic acids, and no outer membrane. So from inside outwards, cytoplasmic membrane and then thick peptidoglycan layer is present. Then gram negative bacteria have a thin peptidoglycan layer, a periplasmic space, an outer membrane, lipopolysaccharide, and porins. So their envelope is more complex. Cytoplasmic membrane, then periplasm with a thin peptidoglycan, and last outer membrane. The most clinically important component of a lipopolysaccharide is lipid A. Lipid A is responsible for the endotoxic activity of a gram negative bacteria. So, even before discussing staining, we can already see that these two bacterial groups have a very different cell envelope structures. But how does this difference produce the gram reaction? So the important concept is the formation of the crystal violet and iodine complex or CVI complex. So after crystal violet is applied, iodine acts as a mordant and forms a larger complex with the primary stain. Now comes the critical step, which is decolorization. This is the step that determines whether the bacterium remains purple or loses the primary stain. To explain this, several theories have been proposed. So for our understanding, we will focus on two important explanations. First one, pH theory, and second one is a cell wall theory. So the first explanation is the pH theory, also called the acid protoplasm theory. According to this theory, the protoplasm of a gram-negative bacteria is considered to be more acidic than that of a gram-negative bacteria. Because crystal violet is a basic dye, the acidic cellular environment was proposed to have a greater affinity for the dye. So this idea was more acidic cytoplasm gives a greater affinity for basic dye and greater stain retention. So, however, this theory does not completely explain the Gram reaction. The modern and more useful explanation focuses mainly on the structure and permeability of the bacterial cell envelope. So, next point is cell wall or permeability theory. This is the most important concept to understand in gram positive bacterium. The cell wall contains a thick peptidoglycan layer. When alcohol or acetone is added during decolorization, the peptidoglycan becomes dehydrated. The structure becomes more compact and the pores become smaller. As a result, the crystal violet iodine complex becomes trapped inside the cell. Therefore, the bacterium remains purple. In gram negative bacteria, the peptidoglycan layer is thin and there is an additional outer membrane rich in lipids. During decolorization, alcohol or acetone affects the lipid rich outer membrane and alters the permeability of the cell envelope. So, the crystal violet iodine complex is then able to escape. The cell becomes colorless when the counter stain, usually safranin, is added. The bacteria take up the counter stain and appear pink or red. So, the central idea is gram positive bacteria retain the crystal violet iodine complex while gram negative bacteria lose it during decolorization. So, the memory trick is positive means thick peptidoglycan, which is traps the crystal violet iodine complex and becomes purple. In gram negative, thin peptidoglycan with outer membrane loses the crystal violet iodine complex and appear pink or red. Next point is modification of gram staining. The original gram staining technique has been modified for different purposes. For this lecture, remember these five different modifications. First, couple of Biermann's modification. In this, primary stain and counter stain used are methyl violet and basic fuchsin respectively. Then second one is a Jensen modification. In this modification is particularly useful in demonstrating meningococca and gonococca. So remember, Jensen means Nigeria. Then the Preston and Morrell's modification. This modification uses an altered staining decolorization. This modification uses an altered staining and decolorization approach to improve control of the gram reaction. Here, iodine acetone used as a decolorizer. Then Biget's modification. This modification is particularly useful for demonstrating bacteria in tissue section. And then the Brown and Brain modification. Brown and Brain is used for mostly actinomycetes. So here we see the clinical significance. The difference between gram positive and gram negative cell envelope is not only important for staining, it also influences permeability, antimicrobial susceptibility, host inflammatory response, and the pathogenic potential. For example, the outer membrane of gram negative bacteria acts as an additional barrier, and lipopolysaccharide, especially lipids, its lipid A component contributes to the powerful inflammatory response associated with the gram negative infections. So here is the bigger message. Cell wall structure determine not only how bacteria stand, but also how they interact with the drugs and the human body. So today's question was, why do gram positive and gram negative bacteria stand differently? The answer lies mainly in their different cell envelope structure and permeability during decolorization. Gram positive bacteria have thick peptidoglycan which retain crystal violet iodine complex and appear in purple. Then gram negative bacteria have thin peptidoglycan plus outer membrane which loses crystal violet iodine complex and appear pink in color. So we also learned two explanations of the gram reaction, pH theory and cell wall theory. And we remember five important modification, couple of Biermann's modification, Jensen modification, Preston and Morrell's, then Wiget's modification and Brown and Brain modification. So now that we understand the structure surrounding the bacterial cell, let's move one layer inwards. Immediately beneath the cell wall lies a structure that controls transports, participates in energy generation, and is essential for bacterial survival. In the next lecture, we will answer what does the bacterial cell membrane do? Thank you for learning with MicroVerse. Let's master microbiology one concept at a time.